let me start you off. This is kind of a fun little diagram here, or a fun picture. I don't know how many of you are familiar with the artist Andy Goldsworthy. He goes out into nature and he makes these incredible kind of monuments of art out of different pieces from nature, maybe logs or different colored leaves. If you haven't looked him up, definitely do it. I would even forgive you for doing it during this talk. One day in that spirit, I had an artist friend of mine come visit our lab. Like what structure would he put together if he just was picking pieces from the lab? So as you can see here, he grabbed filter tip pipettes, he grabbed cryo vials and conical vials, but he also, at my request, grabbed Ethernet cables, right? Other computer cables. And the whole idea of this little piece of installation art, right? And there he is right there, is to take both experimental tools, these vials, these filter tips, and computational components, these cables, and bring them together to build a model of a cell, right? So this is an artistic version of what I'm going to be talking about today. How can we generate representations integrating computational and experimental components to really accelerate how we learn things in this new era of biology? Gaute kindly referred to this work that we've done now getting close to a decade ago, building a computer model of the mycoplasma genitalium cell. I'm going to springboard past that by talking about one of the maybe lesser known challenges that Francis Crick proposed in 1973, which is actually my birth year. So he and Sidney Brenner, and kind of a lightly credited Sidney Brenner, proposed this Project K as building a complete solution of E. coli. The idea was that they put all their eggs into one basket, try as a global community to build a model of E. coli. Same thing, taking as much as they possibly can into account and really solving this bacterium. Proposing a central lab where everybody's generating somewhat straightforward data sets and very integrated. You know, you can imagine, especially, you know, having had chances to interact with Paul Allen, you know, without whom none of this would be possible. You can totally imagine him wanting to set up something kind of like this, whether or not it would be about E. coli, maybe it would be about a neuron or whatever, but just this idea of a complete solution, right? Something that brings together the mathematics, the computation, and this idea of this kind of industrial scale, global approach to getting the experiments all together. Of course, even though there were some attempts, no one ever did anything like this. And so we still see this as kind of our current goal in what we call the E. coli whole cell modeling project, or more affectionately named and with the same acronym, the E. coli whack-a-mole project, just because of everything that's required for us to try to get this thing done. What does it actually mean to build a model like that? What we're trying to do is take all of the data we can find about E. coli, and you can see here year by year, different papers appearing, we pull them out, we're trying to get the data out of them all. We end up with this vast treasure trove of data that's very heterogeneous. I'm just showing you the number of data points that we got from each paper here. How do we integrate all that? The answer is with mathematics, right? So you can imagine writing out thousands of ordinary differential equations governing the expression of all the RNA in the cell, putting that together in mathematical equations. Same thing with the 1,500 or so small molecules that are known and associated with E. coli, wrapping those in equations, and the same thing with the proteins, another 4,500 or so equations. And now we've encapsulated all those data in mathematics. And if we further group them into their functional categories and then use kind of our whole cell modeling secret sauce to put all of this together into one unified whole, we're able to start thinking about really having all of that data together as something that we can represent and that we can even use to run simulations. And just to drill down in that a little bit, you can see here all of the different kinds of data that we're taking into account, right? So they're all color-coded for reasons that'll be clear in a moment, but we take all of these different kinds of data sets, and from them, we extracted over 19,000 parameters. And then we take those parameters and we wrap them in mathematics, as you can see, right? Like here are the equations, and the color coding kind of tells you which data goes into which part of those equations. And I should say here, the ODEs are like a starting basis. Not everything, if something is only known in very qualitative ways, we don't use ODEs to describe it. We have a lot of different types of mathematics. That's a subject for another day, but I'd be thrilled to answer questions about it. So from all of that, we build up this model and we can make these kind of incredibly rich simulations, which kind of have a life of their own and which we honestly kind of think of as its own organism, right, that we study, which we then can project out going back into the real organism. So right now we have, even this is out of date now, but we have 3 times, probably more than 3 times the number of genes included as in our original model. It's much more complicated. It can grow out to arbitrary number of generations. And most importantly, maybe, is that the original model ran on a supercomputer over 10 hours for 1 cell to divide 1 time. Now it runs on a laptop over 10 minutes. So it's a very big difference. Anybody can run these. And here are some example simulations, just some animated output. You can watch E. coli growing in a minimal medium over here. Here's the DNA. Everything here is based on actual simulation data. You can see the mass grows out. When it divides, it reduces back down. So like I said, we can run arbitrary number of generations, which was actually very new, depended on new discoveries made by Suk-Jun Jun, who was also supported by by Paul Allen at some point some years ago. So it was really interesting to see those projects come together as his work enabled us. Something that I also want to show here is that whereas the original model of M. genitalium could only grow in 1 environment, really the only culture environment known for that bacterium, here we can shift to another environment. What you'll see is that a bunch of sensors are going to get triggered, after which genes will turn on and off, metabolism will be rerouted. The net result of all of that will be to increase the ribosome concentration, which leads to the cells growing out longer. They'll be thicker, they'll grow faster, and the DNA, they'll be growing so fast that the DNA has to kind of parallel process here. You see these multiple replication forks forming. It's quite amazing, actually, to watch all this. And this is just one tiny fraction of all of the data that's generated when we run these simulations. Everything that we do right now is available on GitHub. And I encourage you to go there. Like I said, Python, everything, there are people right now using this around the world. COVID was an interesting, it was a very difficult time, obviously, but in terms of this project, was interesting because several labs found themselves with time on their hands, and computational work had a renewed appeal as no one could go into their labs. And so we now truly do have people using this, well outside of the group. And so as a result, I think it's really, hearing their feedback has made this a lot more user-friendly. But the real fun of having a model like this is try to use it to learn something. And in particular, what we want to do is to identify, unanticipated behaviors, large and small, right, that we can actually go in and measure. So I'm going to talk about that a little bit more. And in particular, in the context of how to reconcile a vast set of heterogeneous data. Now, what's funny is, we're talking about, again, think about all this junk on the table from Apollo 13, right? We're trying to fit all of that together. I think what is interesting is that simultaneously there's this crisis, as shown in this Nature article. People are simply worried about reproducibility. Like, is it even the case that we can trust most papers that come out? Like, is it a reproducible finding? And that's a very interesting question on its own. I think it is interesting here. 52% of scientists think this is a significant crisis. We're trying to do something that's, I would say, a whole dimension beyond that, which is trying to say, if you have all of this data, does it give us a self-consistent picture of what we know about the E. coli cell, right? Do all of these things together add up to something comprehensible and consistent? And you can only do that with a large scale model such as this. We call the process deep curation. Deep in this case is intended to reflect multiple layers. Like if you think about deep learning, right, where you look at interactions between things and then you look at interactions between those interactions and so on down these different layers. In our case also, we're thinking about layers of curation. And I think primarily the community has only focused on one layer, which is, so now I'm just to orient you, I'm showing you what I showed you before, but just turning it on its side, primarily just on the data itself, right? Housing it, right? Having a place where it's machine accessible, that's all super important, right? But there are several other layers that are required to be able to compare these data to each other. So the next big layer would be the parameter layer. And I want you to think about, for every parameter, it's representing, let's say, at least 3 and maybe hundreds of points as it's fitting curves or having a model about how those data are related to each other in the context of that experiment. So this parameter layer is derived from the data. And then you have a further layer. The parameters contain the data. The equations contain the parameters. And as I said, you know, that encapsulates not only the data, this is an interesting part, but it also encapsulates the mechanism, right? If you have the equations, it's a second level of curation that has to be performed because you have to know what the relationships are between those parameters, and you incorporate that in this next layer. Then, of course, there's this unified model layer. What I want to emphasize here is that now, once you have this, every single piece of data over here is linked directly to every other piece of data. It just has to go through the layers, right? So a piece of data here might go down and play in this parameter and play in this equation, but now through the context of this, you can go back up through this equation, and now all of a sudden it's connected to this. Right? And so every single piece of data can be cross-validated against all of the others, which is something that's really powerful. When we think about the utility of a model, almost always people think only about prediction. I want to emphasize here that interpretation is at least as important, and especially in our data-rich, knowledge-poor environment, right? This is a, you know, since I'm talking to brain people, I'll bring up that old, obviously untrue adage, you know, the average human only uses 10% of their brain cells. We know that that's not true, but I think we all would agree that the average scientist uses far, far less than that of their high throughput data, right? So that's what we're talking about. How do we get the maximum utilization out of that? And so once you've got this unified model, you have this also really interesting thing, which is that now you have access to millions of data points that were never generated, right? For which no experiment exists, and which are completely, though, accessible. I wrote a whole paper on this relative to weather prediction recently, comparing what's been achieved in weather modeling and Earth systems modeling with what could be achieved in biological modeling. And I think they've really mastered this idea of using the simulation output in innovative ways. Okay, so I'll give you a really simple example of how this works, and then I'm going to try to blast through, being very mindful of my time. I don't want to be the first speaker and make everybody run late today, so I'll try to stay right on time. But I'll give you a quick example of how this worked in the original M. genitalium model, just because it's easy to wrap your head around. So in M. genitalium, one of the things we had to figure out was how much DNA was in a cell. Just think, sounds very simple, right? Like just how much DNA is in a typical cell. And turns out there are two ways to do it, as there often are in these kinds of models. One way was to take a series of reports, right? One which reported the water content of the cell, one which reported the density of the cell, the diameter of the cell. So we had kind of this idea of how much total weight there was. And then from there, a measurement of the mass fraction, okay, which was just a small number, 4% in this case, measured in the 60s. So that gave us a certain number, which is down here. We could just crunch through that and get an estimate. Over here, though, we have a new high throughput, high tech way of doing it, which is just to take the genome sequence itself, right? You can add up the molecular weights of all the bases. When you add them all up, you get the mass of one chromosome by sequence. And when we do that, we get a different number. And look at this. It's kind of just an amazing, but simple, but amazing thing. Here you can see that when we do that, we only have about a fourth of, by this measurement, we only have about a fourth of what would be required to create an entire chromosome, right? So right away, we know that something in here, or we would guess that something in here is probably incorrect. Cell water content, you know, we can't make it 0, we can't make it 100. So there's a range here that's allowable, and it's the same for all of these, right? And so we can go through and quickly identify then that the mass fraction must be the one that's incorrect, and we can make the according adjustment, but an adjustment that actually is a prediction, right? That this measurement probably was incorrect. It's not so different going back to Crick and you know, back Watson and Crick, you know, when they postulated the double helix for the first time, it also was a case in which that model did not fit all of the data, but the model helped to show them which data fit and which potentially did not. And those data did in fact turn out to be incorrect, as the future would tell. This is the same thing, but now think about it as at a massive high throughput scale. Okay, so let's start thinking about deep curation and thinking about it in particular, asking this question: how self-consistent are the data, and where are the points of friction in the data, right? Where it might be the case, like I showed you before, that there are two sets that really don't match. We'll do this quickly, but with 3 different, very different types of data and different kinds of data sets. One old one from the 70s, where it's just simply like the doubling times of E. coli and certain other physiological measurements. One is the metabolic fluxes. It's now over a decade old, and one is more recent, which is a proteome, the amount of every protein. So let's look at the doubling time first. I think that it's complicated, but it gets us started, right? So when we looked at the E. coli simulation at first, we didn't want to change any of the parameters. We just wanted to let them have their say. So we put in those 19,000 parameters. People often think of this as kind of like a neural net or some kind of a trained thing where you might have hundreds of thousands of learned parameters. This is not that at all. This started out as 19,000 parameters, none of which, like all of which, were taken from measurements. And then we tried to see from that what we could learn and what was probably right and what was probably wrong. And so the very first thing that showed up, we had this measurement, as I said, the doubling time, less than 50 minutes. But when we simulated it at first, we had something that was much, much longer. Okay, and here's the range, because there's a significant amount of stochasticity built into our simulations. So the question was, why is that arising, right? And as others have noted, there's actually a beautiful review about this. Trying to model things at this large scale really is going to necessitate some new mathematics, in particular thinking about sensitivity, thinking about just how to analyze and estimate parameters, et cetera. And so one complication here is that everything is so interconnected, it can be difficult to find out who the major players are. I won't go into too much detail about this, but the idea was that instead of doing a single parameter sensitivity sweep, we had to choose groups of parameters at the same time. So we chose a bunch of groups of values to change at random, and always look to see which players, when there was an impact on the growth rate, which players tended to show up in these modified groups, if that makes sense. Again, happy to discuss anything in more detail at any time. So we ended up with this plot in which we could kind of look at the players, the different parameters in particular that tended to always show up when the growth rate was changed. And several of these should not be surprising to you. So these are all ribosome, related to ribosome expression. These over here are related to RNA polymerase expression. There were a couple surprises. I'm going to save that for later in the talk because it's one of my favorite stories. But just so you can see, some of the key players were here: ribosome and RNA polymerase. And so we actually went back and took a look at those. We started making more changes in both ribosome expression and in RNA polymerase expression. And what we found was kind of interesting. You can see that here's our original parameter set. If we change the ribosome expression, excuse me, we see that we can move it. These have closer agreement. If we change the RNA polymerase expression, we can move it, but we really have to change both of them together to be able to match our growth rate. And what was interesting from there was we actually, in doing that, we had far greater expression of both the RNA polymerase and ribosome, and something that actually mapped very well to some new measurements that we consulted later. But so we knew that we were onto something here, but we didn't know why. And that's where the deep curation gets really interesting. I wish I could show you exactly how this works, because it's like a masterwork here. This was done by Hee-Jo Choi in the lab. She goes through and she's made this diagram where you can see everything that goes into our calculation of how much ribosome or RNA polymerase there should be. And you can see that there are 11 different sets of parameters that contribute to this, all that have been measured in one way or another. And so what she decided to do was say, okay, let's go through every single one of those, and let's see, first of all, how many times has that measurement been reported? Like, is it well characterized or maybe a little less well characterized? How much variability is possible? Just like I told you before, there can't be 0% or 100% water concentration in a cell. Some of these parameters are the same way. They're not that flexible in terms of how much we can move them. So she went through and then once you have that, you can also simulate how big of an impact it has, right? And now we know that it should match these RNA polymerase and ribosome numbers, and also should match our doubling time. So when she went through all of these, and look at this, it's just beautiful, right? She could go through for every single one and go see. And when you actually went through, prioritized here and there, I won't go through all these details, but you could see that there was only one set of data that if you changed it would actually lead to this like doubling time correct, ribosome in the right ballpark, and RNA polymerase in the right ballpark, all with regard to the actual experimental measurements. And so we were able to quickly identify mRNA expression as the most problematic of all of these measurements, which was fascinating, right? So we were able to go through and take a closer look at some of those measurements, which has been extremely helpful. And as a result, we had a simulation that fit better. So this is a relatively kind of a first pass example, but hopefully it gives you this idea of the complexity involved in this kind of a deep curation process, and all of the data that has to come into a thing like this, and all of the simulation and equations that have to go into something like this. And you can see here again, how we think about this in terms of this deep curation, like where everything fits into that schema. Okay, so hopefully that's illustrative. And now I want to talk a little bit more about a more interesting data set, the metabolic data set. Just check the time here. Okay, great. And I'll go through this one fairly quickly. So here we've compared our simulations to the flux measurements, and what we see, here's now what was measured versus what was simulated. And you can see that in most cases, there's a great correlation there. But there were a couple of major outliers, right? Really major outliers. And so the question there was, what's causing that? And I think a natural tendency would be to say, oh, we need to fix parameters connected directly with this. But metabolism is like on the order of 1,000 enzyme network, right? And all kinds of things could be changing this. And so we had to do a more systematic approach. We did something like what I told you before with the growth rates. We found a set of, let's see, 13 possible candidates that we evaluated in more detail. And we ended up with a smaller list of 8 that really seemed like very important. And so then we were able to do something you would never consider doing experimentally. We could do a full factorial experimental design where we ran all these sets of simulation runs. And keep in mind that 256 sets of simulation runs means that we're running, multiple different random seeds, running out each one for many generations, trying to see what impact that has on all of that together. So it's pretty extensive it's a pretty massive experiment. Even simulation wise, you would never do it experimentally. So what we're trying to see is if we remove the impact of certain parameters, does that give us the opportunity to grow in a normal way? I won't go through in too much detail just for time's sake, but we were looking for, in particular, modifications that we could make that would give us a similar, like a nice correlation with the validation, the metabolic fluxome, but at the same time would keep us in this correct growth regime, which we're noting here by the yield. So these orange points are the ones that kind of fit the bill, and they all have the same four, they all basically are pointing to four enzymes in particular for which the parameters are thought to be wrong. Changing them, as was suggested, leads us to something that has a much better correlation. But more importantly, I think here we end up with kind of, here is our original estimate of around where the kinetic parameter should be based on data that existed previously. And with our new model approaches, you can see that we always are arriving in this better spot, especially when it comes to these newer measurements that are created. Like, the model is amazingly, to me, able to generate these new predictions about where specific parameters should lie in this vast network and get them right, which is just incredible. I won't talk too much about this, except for just to say that in the end, the entire network is remarkably self-consistent. These things down here at the bottom, as I told you before, this is not a whole cell model yet, right? This is a half cell model, is how we affectionately refer to it. And these things down here represent bridges to other parts of the cell that we haven't yet included. I'll tell you a last thing here. I see that I've got to make sure I'm going quickly to be on time. We compared everything to the proteome. And so you can see here again, this is measurements of all the proteins in the cell, protein levels. Here's what we simulated. It's, again, a very nice correlation, but not perfect. And I want to just illustrate a couple of things that came up that I thought were super exciting. When we looked at this list, as I told you, we found the ribosomes and the RNA polymerases, but we also found some surprises. And maybe the biggest one to me was this cdsA, which is a membrane biosynthesis protein that I just wasn't expecting to see there. When we started looking at what was going on, here's the model. So the model has a very low level of cdsA. And so you can see what it does is it leads several simulations to fail because there's this long tail of very low phospholipid biosynthesis, okay? And if we were to bump that up, you have a much more robust simulation. The problem is when we went in and actually did the qPCR, we didn't see, like, it looks low, right? It looks very low. And it got kind of weirder. You know, we looked at ours, we said it was low or no expression. We looked at the protein, the proteome there that I just showed. It gives a low but non-zero amount of protein. But then these two papers independently say that it is essential for the cell. So that was kind of an interesting thing. How can something that's not really getting expressed be essential? We ended up doing a study here where I'll skip the details, but basically it was a steady state analysis in which you would assume that the production rate of each protein and the loss rate should be roughly equal for cells growing in a log culture. And what you see is that, again, most proteins fall into this mix, but there are some exceptions, right? There are some exceptions. And those exceptions are probably, there's something that needs to be done with deep curation. Some parameter is not adding up. The parameter that we identified came from a paper in which, you know, it turns out that decay rates of proteins can be difficult to measure, and so there's this thing called the end rule, which assumes a half-life based on sequence, based on the protein sequence. And in particular, it kind of assumes that some proteins are very stable, like on the 10-hour time frame for their half-life, and some are degraded just after a few minutes, There's not a lot in between there. And so we thought this could be a very likely place for parameters to be updated. So we actually went in, and for some of these outliers, these 6 outliers, we went in and made the measurements. And in all of the cases where the NN rule said long half-life, the model said, no, it should be shorter. In fact, it was shorter. In the cases where the NN rule said a very short half-life, but we said, the model said a very long half-life, the model again was correct. And oftentimes, if you see here, like tens of hours, right, versus 2 minutes, that's a pretty significant difference postulated and then proven by the model. So a very important experimental demonstration. But also I want to note that cdsA, our membrane protein, right, falls right here. And so we did an experiment that normally you wouldn't do, right? A membrane protein is typically a very difficult thing to do a half-life for. We did an immunofluorescence-based assay, and here's a protein where it's known to have a very short half-life. You can see that the fluorescence disappears very quickly. cdsA should look just like that if the data were correct, but it turns out that the model is correct, and in fact, it is much, much longer lived. So this kind of solves our problem of what parameters need to be altered to be able to make the cell grow at the right rate. But it also demonstrates this incredible power of deep curation to give us new insights and tell us about the self-consistency of the data set. So I'm going to tell you, I want to tell you just a couple more things before I quit, or just a very, I want to get very close here to the end. Let's see, I just want to show you one thing, because I know you're over at the Allen, and I love this data, and I think you'll appreciate it. So we're currently, I'm skipping kind of some of our other fun demonstrations, but we are starting to move. You know, my goal is one day to be able to model increasingly complex cells and groups of cells interacting. And I believe that this is eventually going to get us in some very exciting places. Okay, so we've already been working on the simplest bacteria, more complex microbes. I won't have time to show you this, but on these colonies. And I believe we're at this inflection point right now. It's been very challenging to get where we are, but I feel like now with other groups joining, et cetera, we do have a lot of what we need to be thinking about moving forward. And I'll just show 2 things. We're trying to go both up and down in scale. Down in scale with Art Olson, Martina Mariton, and David Goodsell. What they've done is incredible. They're using CellPack, which I think is something that they'll know very well over at the Allen Institute. But look at this. Here is a model in which our whole cell simulations were used to create the data from which they could make a CellPack representation in 3D of all the molecules in the cell. The nice thing about this, to me, what's even more exciting than to look at this is to know also that there were 3 data points out of the simulation that were made. Previously, it's always only been 1 data point, so that's super exciting. The idea of having a dynamic version of this that could go through a whole cell cycle would just be amazing. And then the final thing I want to show you is that, oh, and I should note, Ryan is at the Allen Institute. I don't know if he's here today, but he was in our lab for some great years, and now he's working in the Allen Institute. One thing that he and colleagues were able to do is start to run the first simulations in which many E. coli are interacting together at once, right, in a shared environment. And to me, this is the first step when you see this. These are the first whole colony models, because every single one of those cells is running the whole cell model. This is all on Google Cloud. But the other thing I want you to note is this is the first step to a tumor or to anything else, any other large scale system that we might want to capture with all of its interactions and connections. With that, I will wrap up. I want to thank my group, which is amazing. I want to thank Paul Allen in particular. I think he uniquely had this vision for us and pushed us farther than we thought we would go. And I will be forever grateful for that, and also just for our other funding sources. with that, thanks a lot, and happy to take a question or two.